next speaker is Dr. Sangkyun Cho. Dr. Cho received a BS from Johns Hopkins University and a PhD from University of Pennsylvania. He is currently a postdoctoral fellow at Stanford Cardiovascular Institute under the mentorship of Dr. Joseph Wu. His interest is in leveraging engineering tools and principles to develop new mechanotherapies for cardiovascular diseases. His title today is Selective Inhibition of Matrix Mechanosensing in Stromal Cells, Attenuates Cardiovascular Fibrosis. Please start your talk when you are ready.
Great. Thank you for the kind introduction and for the opportunity to share my work. My name is Sang. I'm a postdoc in Joe Wu's lab at Stanford. And today I'm really excited to tell you a little bit about my work that focuses on selective inhibition of matrix mechanosensing in stromal cells for the development of antifibrotic therapies. And so for this audience, I don't think I need to go over too much detail, but briefly, fibrosis, as you know, is a key component of a vast range of cardiovascular diseases. In the case of the heart, it's simultaneously a consequence as well as a contributor to many different pathological conditions as well as aging. I mean, despite being one of the key indicators of adverse patient outcomes, there are no therapies available. And depending on the physiological or molecular insult, you can get many different types and shapes of fibrosis forming in the heart. For example, you can get peravascular fibrosis around blood vessels, interstitial fibrosis in between cells, and then, of course, replacement fibrosis when you have things like myocardial infarction. But one common denominator of most, if not all fibrotic hearts, kind of by definition, is mechanical stiffening, right? So, for example, a normal adult heart falls in the range of about 10 kilopascals to 20 kilopascals in terms of stiffness or microelasticity. But then when you have diseases like MI, HFpEF, or pressure overload, the myocardium can stiffen up to 5 to 6-fold. And as you know, fibrosis occurs when you have quiescent cardiac fibroblasts in the myocardium exposed to a range of pro-inflammatory soluble signals like TGF-beta, PDGFs, angiotensin II, so on and so forth. And these signals activate these fibroblasts, and they transdifferentiate into a distinct cell state, or some people might argue a different cell type called myofibroblasts, which are the culprit cell types that are thought to remodel the matrix and stiffen the extracellular environment. But what's often overlooked here is that this fibrotic stiffening itself is a well-known activation signal for fibroblasts that promotes even more generation of these myofibroblasts. And so, for example, if you culture primary or iPSC-derived cardiac fibroblasts on hydrogels of varying stiffness, you'll see that even without the addition of these soluble factors, they spontaneously transdifferentiate over several passages into these alpha SMA positive myofibroblasts-like cells, unless you keep the cells in a very soft, malleable environment that mimics that of the embryonic heart, or unless you add in drugs that inhibit Mechano-transduction, the so-called mechanosuppressor drugs, including blebbistatin, ROCK inhibitors, or YAP inhibitors. And so if you also stain for YAPTAZ, which is one of the well-established mechanotransducers in the cell, and quantify its nuclear localization, you see that it's very tightly correlated with the fraction of alpha SMA positive cells. And if you zoom in a little closely, both plots show a characteristic inflection point that occurs at around 12 kilopascals, which corresponds roughly to the stiffness of strided adult muscle tissue. And so if you actually plot these two together, you'll see that, again, the alpha SMA positive cells and the nuclear YAP show robust correlations going from soft substrates to stiff substrates. And here, for example, TGF-beta sort of pushes the curve upwards, whereas if you add in drugs like SB431542, which is a TGF-beta inhibitor, it sort of suppresses the curve and pulls it downwards. And then the addition of these different mechanosuppressor drugs really sort of limits the data points to this bottom left corner here, which suggests that this inherent mechanosensitivity is really important for this spontaneous activation of fibroblasts, and it's driven, at least in part, by YAP-TAZ. And so ultimately what we get is this characteristic positive feedback loop where the stiffened myocardium actually feeds back into activating even more fibroblasts and creating even more myofibroblasts. And this presents many challenges because this feedback loop can be perpetuated even after the initial profibrotic soluble signals have subsided, for example, including IL-1 beta, right? And so as you know, there's been many efforts to develop antifibrotic drugs that target these pro-inflammatory, profibrotic soluble signals, but very few have shown real promise in the clinic. And perhaps one of the many possible reasons for this is because we've only looked at one side of the story. And maybe we need to target not just these soluble profibrotic signals, but also these insoluble mechanical signals to try to create some synergy to suppress or maybe even reverse fibroblast activation. And so to test this idea, we work with cardiac-specific fibroblasts differentiated from iPSCs. And these cells exhibit characteristic morphologies, functional properties, and transcriptomic profiles of primary fibroblasts. And importantly, they have this ability to readily transdifferentiate into these alpha SMA positive myofibroblasts. And so to characterize this a little more, we did some single-cell sequencing. And as you might expect, the response to TGF-beta stimulation is very heterogeneous and dynamic, with multiple clusters that all seem to branch out from this central green cluster here, which we named QCF1, which seems to represent quiescent cardiac fibroblasts. We also have a few clusters that are representing actively cycling or proliferating fibroblasts, a few that are sort of activated and are in transit, and then a few clusters that seem to represent a myofibroblast-like phenotype with upregulation of many of these canonical fibrosis-associated genes, including matrix genes, cross-linkers, and contractility-associated genes. And the goal here was to see if we can actually suppress or maybe even reverse myofibroblast activation. And so we teamed up with Helen Blau's lab at Stanford to use their new dynamically softening phototunable hydrogel system. It's a cool system that starts out stiff like a fibrotic tissue, but then in response to light, 4 or 5 nanometer light, the crosslinks get degraded and it softens in real time on demand. And so you can really observe cells' responses to changing matrix mechanics in real time. And so we use this in combination with a reporter iPSC line that expresses CFP tagged SM22 alpha or transgelin. It's one of the major genes that go up with myofibroblast activation. And we first made these into myofibroblasts and then exposed them to dynamic softening, either with or without soluble inhibitors of the TGF-beta pathway. And looking at the 4 conditions, what we noticed was that only in the combination treatment, where we combined dynamic softening with soluble inhibitors of TGF-beta, do we see dramatic changes in morphology, as well as a gradual decrease in CFP signal. So this is really interesting to us. So we zoomed in a little bit with immunofluorescence to compare the non-softened stiff control versus the softening plus TGF-beta inhibition treatment, and we found that the combination treatment results in a significant decrease in nuclear YAP, as well as a global reduction in alpha SMA expression. And most importantly, we saw a loss of alpha SMA from stress fibers, which is one of the major hallmarks of myofibroblasts. And so we went back to the single-cell sequencing data and compared, again, the stiff control to the softening plus TGF-beta inhibition treatment in orange, and found that there's roughly a 40% redistribution of the population towards this quiescent fibroblast cluster here in the middle. So looking at it a little more quantitatively in terms of the fraction of the total population, we saw that all of the myofibroblast clusters identified sort of decreased in size, whereas all of the fibroblast clusters identified increased in size, most notably this, again, the quiescent cardiac fibroblast cluster here in the middle, which almost doubles in terms of cell number. And importantly, if you look at the expression of SM22 alpha, which was the reporter that we used for the time-lapse images, we saw that the softening plus TGF-beta inhibition leads to a decrease in levels that's almost identical to levels in the non-treated quiescent cardiac fibroblast population, suggesting that it's very consistent with our time-lapse imaging of the dynamic softening experiments. And so next, we wanted to really dive into what's exactly driving these population shifts. And so we did some transcription factor activity fingerprinting by a package called Dorothy. This is basically an algorithm that predicts the activity, not expression, of transcription factors based on all of their known downstream target genes. And so this is a heat map showing the top 25 variable TFs in the quiescent fibroblast cluster and the most prominent myofibroblast cluster with the two conditions. And among the top 25, the top 5 turned out to be the TED family and the SMAD family of transcriptional regulators. As you know, TEDs are co-regulators of the YAP-TAZ and HIPPO pathway, and SMADs are, of course, the downstream effectors of the TGF-beta pathway, so it makes sense that those are the most variable. And these turned out to be most activated in the myofibroblast clusters compared to the quiescent fibroblast clusters, but at the same time, ones that were most heavily deactivated with the softening plus TGF-beta inhibition treatment. We also did some Venn diagram type analysis to look at TFs that were uniquely variable in the softening plus TGF-beta inhibition combo treatment versus the softening only or the TGF-beta inhibition only, and identify 24 TFs that were uniquely variable in the combo treatment. And among the 24, the top ranked one by far was SRF, serum response factor, which, as you know, is one of the master regulators of myofibroblast and smooth muscle cell differentiation. And so looking at this a little closely with SRF cofactors like MKL1 and its downstream target genes, again, we saw that only in the combination treatment do we see global repression of all of these genes, whereas the single treatments had much more inconsistent effects. I mean, obviously, this was all single-cell sequencing data, so we also wanted to validate at the protein level. So we did some Westerns and also immunofluorescence to show that key fibrosis-associated genes like alpha SMA, collagen-1, periosteinol decrease, concomitant with a slight increase in phospho-YAP, which is an indicator of its inactivity. And then, of course, we did some histogram-type analysis with immunofluorescence, which also showed that there's a massive suppression of alpha SMA positive cells that were not seen with softening only or the TGF-beta inhibition only. So the current working model is, we think of TGF-beta being sort of the main driver of fibroblast activation and fibrosis in general. There is this characteristic mechanical feedback loop that's driven at least in part by YAP-TAZ. If you treat these fibroblasts or myofibroblasts with soluble inhibitors of the TGF-beta pathway, it has very modest effects, presumably because of this inherent mechanosensitivity. And likewise, if you just suppress mechano-transduction, either with matrix softening or with drugs, it has very limited effects. And surprisingly, when you combine the two, there seems to be some level of synergy that is driven at least in part by SRF and MRTF that suppresses or even potentiates reversal of fibroblast activation. And so the next question we wanted to ask was, if this is all true, then how can we actually get the cells to perceive a stiff fibrotic environment as soft in vivo? And more importantly, how can we actually achieve this selectively in fibroblasts and stromal cells while not affecting the other key cell types of the heart? And so when you think about different ways to translate to this clinic, I think there's broadly two different strategies. One would be to try to directly reduce the stiffness of the cardiac ECM, but obviously this carries a lot of risk because you don't want to fiddle with a dynamically beating, mechanically stressed pump like the heart. And so I think the more sensible strategy would be to try to target the downstream mechanosensory pathways, preferably specifically in fibroblasts. And so now there are many, many different small molecules that you can explore along the mechanosensory apparatus of the cell. And so I looked at each and every one of these, and one in particular caught my attention, and it was SRC or sarc for two reasons. One, because it's a well-known upstream regulator of YAP-TAZ mechano-transduction. But secondly, much more importantly, we did a meta-analysis of about 6 public single-cell RNA sequencing data sets and found found that sarc is actually highly enriched in fibroblasts and stromal populations of the cell. So for example, this is the human heart cell atlas that was published a few years ago. You'll see that sarc expression is much higher in fibroblasts and pericytes, whereas it's barely detectable in other cell types like cardiomyocytes. And this is true not just for adult human hearts, but also human fetal hearts and several mouse adult heart data sets. So its baseline expression is much higher than in cardiomyocytes or endothelial cells, for example. But more importantly, it's actually activated very, very strongly in fibrotic hearts. So for example, looking at TAC hearts at 2 different time points, you see progressive fibrosis. And along with progressive fibrosis, you see progressive activation of sarc, almost an 8-fold activation by 6 weeks, as assessed by phosphorylation at tyrosine 416. And so this suggested to us that while we can't soften the matrix in vivo directly, perhaps we can use sarc as a fibroblast or stroma-enriched mechanosensor that we could target pharmacologically to mimic the effects of matrix softening in vivo. And so we worked with folks over at Greenstone Biosciences to perform an in silico virtual screen as well as MD simulations to identify this relatively understudied drug called saracatinib as the top candidate, which is capable of penetrating deep into the binding pocket with the most stable interactions. We performed also some connectivity map analysis offered by the Broad Institute. This is basically an algorithm that compares pharmacological perturbations to genetic perturbations. So basically comparing drug treatments to knockdowns or overexpressions of specific genes. And doing this for saracatinib revealed that among the top ranked gene perturbations were, for example, knockdowns of focal lesion associated genes like paxillin, and importantly, knockdown of YAP1, which is consistent with the drug's predicted inhibition of sarc-mediated YAP-TAZ mechano-transduction. And so with this drug, we wanted to test the effects either alone or in combination with 2 different soluble inhibitors of the TGF-beta pathway. And so we did this with iPSC-derived cardiac fibroblasts, stimulated with TGF-beta at 2 different time points, and then imaged alpha SMA, YAP1, and MRTF, which is a co-regulator of the SRF pathway. It's a busy slide, but basically suffice it to say that the combination treatments shown in orange, as well as a reversal of cell morphology back towards a more spindle-like shape, which is very characteristic of quiescent unactivated fibroblasts. And most importantly, when you quantify both YAP and MRTF nuclear localization, only the combination treatments resulted in significant decreases in both nuclear YAP and nuclear MRTF, which is consistent with what we saw with our single-cell sequencing data showing unique downregulation of SRF in the combination treatments only. And then, of course, we also did some Westerns to confirm this at the protein level, looking at key proteins like alpha SMA, collagen-1, periostin, and of course, an increase in phosphorylated YAP. And even at the metabolic level, we saw that the combination treatment shown in orange really reverses metabolic states back towards levels comparable to unactivated quiescent cardiac fibroblasts. And then finally, we tested the effects of this 2-hit combo treatment in a more physiologically relevant model using 3D engineered heart tissues. As you know, these are tissues that beat in between flexible silicon posts. And if you expose them to TGF-beta, within several hours, they undergo dynamic tissue compaction, seen with a reduction in contractile function as well. And surprisingly, so we treat the EHTs with TGF-beta for 2 days and then subsequently added in the combination of the 2 drugs, saracatinib plus 2 different TGF-beta inhibitors, and found that surprisingly it results in almost total decompaction of the tissue as assessed by diastolic length. So in other words, how long the tissue is when it's relaxed. And this was seen with partial recovery in contractile function as assessed by fractional shortening, presumably due to relaxation and softening of the tissue. And then finally, we wanted to test the effects in an animal model. So we went ahead and tried it in the TAC pressure overload-induced hypertrophy and heart failure model. And before actually starting the experiments, we used a public data set of TAC at different time points and first confirmed that the same exact TFs are indeed the ones that are most activated in the TAC hearts at week 5. And so we tested 6 different conditions, sham with just the drugs, and then TAC with 2 different time points, and then TAC followed by the combination treatment, and then TAC followed by the single drug treatments. And we were pleased to see that the combination treatment showed very promising effects in reducing not just interstitial fibrosis, but also perivascular fibrosis, whereas the single drug treatments had much more modest effects. And most importantly, we saw a partial recovery in systolic function as assessed by LVEF at week 6. And finally, we wanted to just confirm that the same molecular culprits are sort of driving these changes. And so we did some Westerns to look at phosphorylated sarc, for example, and saw that the combination treatment results in almost near abolishment of sarc activity, coinciding with decreases in periostin and alpha SMA in these hearts. So just a quick graphical summary. Again, we think of TGF-beta as being sort of the master regulator of fibroblast activation and myofibroblast transdifferentiation. What's commonly overlooked is that there is this characteristic mechanical feedback loop that's driven in part through YAPTAZ. We identified sarc as a fibroblast-enriched mechanosensor that we could potentially target to mimic the effects of matrix softening in vivo. We demonstrated that treating hearts or fibroblasts with soluble compounds that inhibit TGF-beta has very modest effects. And likewise, softening or saracatinib treatment alone has very limited effects in suppressing fibroblast activation. But interestingly, when combined, these create synergistic effects driven at least in part through MRTF and SRF to shut down profibrotic gene programs and potentially reverse fibroblast activation. And so I'll just quickly just go over highlights and conclusions. I'll just mention that, again, sarc is a stroma-enriched mechanosensor, and this is sort of a first proof of concept mechanotherapy for treating cardiovascular fibrosis. And so with that, I'd like to thank everyone who helped out with this project in the Wu Lab, especially Xian, who helped out a bunch with the mouse animal studies, our collaborators in the Helen Blau Lab and Greenstone Bio, and then, of course, our funding sources. And thank you, and I'd like to take any questions if you have.
Okay, great talk. Does anybody have any comments or questions? I think you showed after TGF-beta treatment in single-cell RNA sequencing, you showed 8 clusters of cardiac fibroblast.
In single-cell RNA sequencing?
Yeah, in single-cell RNA sequencing. Mm-hmm. And you showed the myofibroblast go back by the combination treatment. What happens in the remaining 7 clusters?
Right, that's an excellent question. So I think there's been several studies in the literature that suggests there's like a narrow window in the timeline of fibroblast activation, after which, if you pass that window, it's going to be basically impossible to reverse myofibroblast differentiation. And so, I mean, I guess you never know for sure, but my first guess would be that the remaining 60 to 70% of cells that do not seem to revert back are those cells that maybe have passed that critical threshold and are maybe too, quote unquote, terminally differentiated.
They are kind of myofibroblast or still quiescent fibroblast?
I would assume that the ones that didn't revert are myofibroblast-like cells.
Myofibroblast-like
or maybe even cells that have transdifferentiated into an entirely different cell type. But there's no way to know for sure with just this experiment. But
any questions? Ionis.
I guess more of a technical question. Is there an advantage of using these induced pluripotent stem cell derived cardiac fibroblasts-like cells versus, say, get fibroblasts from a healthy patient to do all this work? Or
right. You can use just regular primary fibroblasts, and in fact, that's probably a better option. But I think one merit is that we can use reporter systems, right? So, for example, this time-lapse imaging study, you can actually observe cells in real time, right, with the reporter, how they respond to dynamic softening. And this provides a lot of advantages because if you just take cells cultured on plastic and then lift them off and then put them on a soft gel, there's all these confounding effects of trypsinization and replating that you can't really account for. And so if you want to try something with live reporters, first.
Have you compared, say, primary human cardiac fibroblasts with you get the same distribution of populations?
Right, we get roughly the same distribution doing sort of these histogram type analyses with primary versus iPSC-derived fibroblasts. Yes.
Thank you.
Any questions?
Great talk. I just have a question regarding to your the screened sarc inhibitor. Have you ever tested whether giving the drug to either iPS-derived cardiomyocytes or in sham mice treated with this inhibitor would affect the cardiac function, like contractility or even like arrhythmic events? If you ever checked that out.
Excellent question. So we did treat the sarcatinib drug to cardiomyocytes and endothelial cells, or iPSC-derived cardiomyocytes and endothelial cells, and looked at viability and their function. So we did like 2 formation assays and contractile function, assess contractile function, and we didn't see much change, at least until about 10 micromolar, 5, 10 micromolar range. But yeah, we can't really deny potential side effects on other cell types, even if sarc expression is low in these other cell types. And that's sort of the direction we're trying to pitch this, but yeah. And regarding the mouse studies, we've also collected the blood in all of these animals. And so if there's some potential toxicity, not just in the heart and other organs, we might be able to detect that through analyzing the blood and serum.
Any other questions? I have a question about the mouse part. So I think you showed the improvement of ejection fraction. I think people believe fibrosis is a kind of consequence of cardiac damage. So can you elaborate how inhibiting fibrosis can improve cardiac function globally?
Right. I mean, short answer is it's unclear at the moment whether there's a definitive reversal in cardiac function and total recovery. And I think the first thing that comes to mind is maybe there's increased matrix turnover and a reduction in fibrosis, so there's less mechanical resistance for the heart to beat against. And there's some preliminary data that I've generated that suggests that treatment with this drug or inhibition of Mechano-transduction actually leads to this rare decoupling between matrix synthesis and degradation. So typically these two go hand in hand, and this is a major hurdle for a lot of fibrosis-focused projects. But what happens when you inhibit matrix Mechano-transduction is that expression of MMPs shoots up while expression of collagens and cross-linkers stay down. And that's typically not the case for many of different antifibrotic drug treatments. And so I think there might be some clues there, but it's definitely something we need to sort out.
Thank you. One more question.
Yeah, maybe. I wanted to ask you whether, do you think that these two different pathways related to TGF-beta and the stiffness, are they converging also to the same gene regulatory networks, meaning that the SMADs and the YAP would actually bind together at these regions, or they're activating an independent set that then are providing this increased effect that you have in protection? How do you see that, and what you're planning to do to maybe go after this question if interesting for you?
Yeah, this is an excellent question, and like you said, this is something I've been thinking about, and I hope to be able to pursue in the future. There's been actually several studies in the past that look at these context-dependent signaling and context-dependent crosstalk among these pathways, most notably TGF-beta and YAP-TAZ. And obviously, there's some evidence in the literature that suggests that they might interact with each other either directly or indirectly. To be honest, at this point, I don't know exactly what their contributions are, how they crosstalk, but at least with our data, it's evident that one is not enough. For some reason, you have to shut down both. And There might be some crosstalk still there, but the main point is that you gotta hit both. If you leave the other, it might cross over into the other pathway.
Thank you. No question. Okay. Then we will move on to the next speaker.